Before going into mitosis, let's discuss about interphase. Interphase is the phase that happens before mitosis, where cells continue to grow and duplicate their organelles. This is the phase where cells prepare themselves for cell division. Interphase is divided into three stages. The first stage is called G1, the second stage is called S, and the third stage is called G2. Let's see what happens in the G1 stage. G1 stage occurs immediately after mitosis. So the cell enters the first stage of interphase. This is where the cell starts to grow and develop its organelles. While developing, it will also replicate and duplicate its organelles. You see, organelles such as ribosomes, mitochondria, endoplasmic reticulum, these things will start to replicate and also develop in. This development of cell organelles will occur in G1, as I said, and then the cell will proceed to S phase. S stands for synthesis. This is the phase where proteins necessary for chromosome formation will be synthesized. While doing so, the cell will also duplicate the DNA. It will double the genetic material, it will double the existing DNA. Why it needs to double? You see, the end result of mitosis is going to be two diploid cells. So the one single cell has to supply enough genetic material for two different cells. So that is why the cell is duplicating its DNA. So that one part of the DNA will be sent to one daughter cell, the other part of the DNA will be sent to another daughter cell. So that is why the DNA is duplicated. Cell has grown, the genetic material has been doubled, cell organelles have developed and replicated, essential proteins have been synthesized. So the cell finally moves to G2 stage, which is the final stage of interphase. So in this stage, the cell looks for error. You see, after finishing exam, after tying the paper, we'll check for errors, right, before submitting the paper. Similarly, the cell wants to ensure that there are no mistakes in the genetic material because it has been copied. The genetic material has been duplicated, so it wants to make sure that the duplicated genetic material has no error. So it will look for errors. If there are any errors, the cell will repair that mistake or error in the genetic material. After the inspection that happens in G2 stage, so if the errors are rectified, if no errors are found, the cell will be certified for mitosis and it will proceed to mitosis from the interphase by the end of G2 stage. Mitosis is divided into five distinct phases. The first phase is called prophase, the second phase is called prometaphase, the third phase is called metaphase, the fourth phase is called anaphase, and the fifth phase is called telophase. Let's see what happens in the prophase, which is the first phase of mitosis. So in prophase, the loosely bound genetic material inside the nucleus, it will be in chromatin structure. I mean, there won't be any structure at all. It will be like a hairball without any distinct structure. It will be like a messy noodle kind of thing. So this genetic material will start to condense. The DNA along with the proteins will start to condense to form the thread-like structure, which we call chromosomes. So the chromosomes will start to condense in the prophase. Okay, and the nucleus, it will have a boundary kind of thing which we call as a nucleus envelope, and this thing, nuclear envelope, it will start to break and it will disappear during this prophase. So the wall is broken, the nucleus wall is broken, and the nucleus will start to disintegrate. Okay, this happens in the prophase. And spindle fibers, you remember centrosomes? So from some centrosomes, a string-like structure which we call spindle fiber, mitotic spindle fiber. So this thing, it's made up of protein. Okay, this fiber will start to appear. This fiber's essential function is to pull the chromosomes apart. So that is why this spindle fiber start to emerge from the centrosomes. All of this happens in the prophase of mitosis. The second phase of mitosis is prometaphase. In prometaphase, the chromosome that started to condense in prophase will still continue to condense, and a protein complex called kinetochore will start to appear at the centromere of chromosomes. So these kinetochores are important because the spindle fiber, the microtubules that extend from the centrosomes, will try to find these kinetochores, because these microtubules will try to attach themselves to these kinetochores. So the centrosome will also go to opposite poles. Two centrosomes will go to the opposite poles of the cell. So all these things happen in prometaphase. After prometaphase, the cell enters metaphase. Metaphase is the third phase of mitosis, and it is very important because the chromosomes that started to condense in prophase will reach its highly condensed state in the metaphase. In other words, the chromosomes are visible, the best visible stage of chromosome is metaphase. You see, that is why these karyograms are taken during the metaphase. In metaphase, the chromosomes, the highly condensed chromosomes, will line up in a place called a metaphase plate, which is nothing but the equator of the cell. The middle place of the cell, these chromosomes will line up like schoolboys lining up for school assembly or prayer. Like that, the chromosomes will line up. So after metaphase, the fourth phase of mitotic cell division, which we call anaphase. This anaphase is the phase where the sister chromatids get pulled apart. So the centromere holds the two sister chromatids together with the help of a protein called cohesin. So this cohesin will break down during anaphase, means that the centromere is no longer holding the two sister chromatids, and the two sister chromatids are being pulled apart towards the different poles of the cell. One will be going to the north, another one will be going to the south pole. Non-kinetochore fibers, the microtubules that are not attached to the kinetochore, it will elongate the cell. The cell will be widened by the end of anaphase, and the cell division will go to the next phase called telophase, which is the final phase of mitosis. In telophase, the chromosomes that the sister chromatids that have been pulled apart in the anaphase will reach the polar region. Half of the sister chromatids will reach the left, half of the sister chromatids will reach the other side. Okay. Remember, chromosomes are counted by the number of centromeres, not by the number of sister chromatids. When the chromosomes appeared in the prophase, they started to appear as sister chromatids, not as a single chromosome. Remember why? Because in the S phase of the interphase, the genetic material duplicated, it doubled that time itself. So when the chromosome started to appear in prophase, it already been duplicated before entering into cell division. So that is why when the chromosomes start to appear, they appear with the sister chromatid. They appear as conjoined twins, not as an individual person. And now in telophase, they have become individual chromosomes and they have reached the polar sides. Remember, the sister chromatids are identical to each other, means they are the same genetic material, and this has reached this side and that has reached that side, means the genetic material has been divided equally to the left and right. Okay, and the nuclear envelope that disintegrated during the prophase will start to develop in the telophase, means two nuclear envelope will develop. Two nucleus will start to develop within the same cell in the telophase. And there's a mitotic spindle, that spindle fiber microtubule. It will break down. Its purpose has been served and now it has no function, so it will break down, it will disintegrate. Each of the sister chromatids that went to the polar sides of the cell will now act as an individual chromosome. So now they will also decondense back to their noodle structure. By the end of telophase, the cell will divide by developing a furrow. This cleavage that happened will depress further, dividing the cell into two different cells. This process is known as cytokinesis. The cytoplasm of the mother cell will be distributed to the daughter cell, and all the physical connection between the daughter cells will be cut off. By the end of cytokinesis, there will be two different cells, two individual cells. There will not be any physical connection between them. After cytokinesis, the newly formed cell will again go through interphase. So it will again grow, it will replicate its organ, it will duplicate its genetic material, it will synthesize protein, again it will prepare for another mitosis, and again it will split into two cells. So this mitosis is happening as we speak. This mitosis is really essential for growth, development, and healing of eukaryotic organism, especially the multicellular organism needs mitosis, because mitosis is a mode of reproduction for single cellular eukaryotic organisms, whereas for the multicellular organisms it is the mode of growth. You remember, we were just a diploid cell. When we were fused by two haploid cells, we were just one single entity of 46 chromosomes, and that zygote divided further, underwent mitosis after mitosis, producing a complex organism with the trillions of cells. So that was possible only because of cell division. This is the end of mitosis. What we saw in mitosis is one cell, one diploid cell, is trying to divide itself into two different diploid cell through the process of mitosis. Okay, it is called binary division because two cells are divided. So it's binary division, and it is also equivalent division because the genetic material and the cytoplasm and the cell organelles are equally distributed. So that is why it's called equivalent division, this mitosis is. So mitosis goal is to create two identical twin cells, two identical twin daughter cells. But meiosis, whereas it differs from mitosis, because meiosis aim is to create haploid gamete, not to create a diploid cell, but to create a haploid cell, which we call gamete. So it has to create gamete, and the gamete must not be same as the mother cell, because it needs to produce variation. You remember, the parents, they look different from their children, means the children looks different from the parents, and these variation occurs because of meiosis. Meiosis wants to create variation based on the existing genes that are present inside the mother cell. So the mother cell will try to produce as much as daughter cells with variation, and it will reduce the number of chromosomes to 23 in case of humans. From 46 to 23 it will reduce, and it will produce gametes. If it is a man, the man will have sperms. If it is a woman, the woman will have egg cell. So these are gametes, eggs and sperms. So the production of gametes happens by meiosis. So let's see what happens in meiosis. Meiosis is divided into two stages, meiosis 1 and meiosis 2. So we'll see what happens as meiosis 1 first. So a single germ cell, a single primordial germ cell with 46 chromosomes. So where these primordial germ cells will be found? If it is a man, it will be found in the male testicles. If it is a woman, it will be found inside the ovaries. So ovaries is the female version of testicles. Okay, it will be present inside. It's not displayed outside like the man's testicle. Um, so we also call these testicles and ovaries as gonads. Okay. So inside the gonads, we have a germ cell, primordial germ cell. It's a diploid cell with 46 chromosomes. And this diploid cell will start to undergo meiosis. So the first stage of meiosis, the first meiosis stage, prophase 1. We call prophase 1 metaphase 1, prophase 2, metaphase 2, like that. We'll add number 1. 1 means it is happening in meiosis 1. If it is prophase 2 means it is happening in meiosis 2. Meiosis 1 consists of 4 stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. So all these 4 stages comprises meiosis 1. And the prophase 1 is the longest stage of meiosis, because a beautiful thing happens in this prophase. So that is why the prophase 1 of meiosis is subdivided into 5 subphases. So these subphases are namely leptotin, zygotin, pachytin, diplotin, and diakinesis stage. So there are 5 substages that happens in prophase 1. In the leptotin stage, the chromosome will start to condense. Okay, it will start to become visible as fine threads with the chromatids. Already the cell has underwent DNA duplication. That is why the chromosome is appearing with sister chromatids. So this happens in leptotin stage. The chromosomes will slowly start to appear. And in the zygotin stage, the zygotin stage of prophase 1, this homologous chromosomes will begin to pair. So what is homologous chromosome and where are they? You see, we know that chromosomes come as pairs. So there are 23 pairs of chromosome inside human body. Inside every single cell, inside the nucleus, we'll have 23 pairs of chromosome, 46 in number. So if there are 46 means we have 23 pair. And say for example chromosome number 1 will have a pair, a similar looking chromosome with similar genes, but they will have different variation. Variants of gene they will have, but they are pair, means they are like husband and wife. And these homologous chromosomes, one set of homologous chromosome will get from father, one set of homologous chromosome will get from mother. So that is why we have 23 pairs. Sperm carried 23 chromosomes and the ovum carried 23 chromosomes. When the cell fused become a zygote, it had 46 chromosomes, 23 pair from mother, 23 pair from father. So that is why we have 46 chromosome. And this 46, as I said, are pairs. Okay, these pairs are called as homologous pairs, and these pairs will start to pair. Unlike the mitosis prophase, where the chromosomes will start to appear, here once they appear along with their pair. Okay, and this appearance is observed in the zygotin stage. This pairing up of homologous pairs is called as synapsis. So far we have seen the 2 substages of prophase 1 of meiosis 1. So the first stage was leptotin, where the chromosome started to become visible, and the second stage was zygotin stage, where the synapsis happened. The homologous pair started to pair. Homologous pair of chromosomes started to pair, and the third stage is pachytin stage. So now the paired homologous chromosomes will start to hug each other, will cross over. They will hug and they'll transfer genetic materials from one side to another. This transferring of genetic material, this process, it is called crossing over. Okay, This crossing over happens in Pachytene stage. So why they should cross over? Remember, as I said, this ultimate goal of meiosis is to create variation. So each of the cell will be different from the actual primordial germ cell, because the genetic material from the homologous pairs will be mixed up, creating around trillions of possibilities. Okay, trillions of possibilities will come uh, through this process of crossing over. So once this crossing over happens, the homologous pair will detach themselves. Okay, wherever this homologous pair touch, this physical touch, this touching place is called as chiasma. Okay, if this arm of chromosome touches that arm of the other homologous chromosome, this place where they touched is called chiasma. Okay, by the end of Pachytene, they will transfer all the genetic material that are essential. This gene will go that side, that variant gene will go this side, and this continue to happen in crossing over. So once this happen, they will detach themselves. The homologous pair will detach themselves, and this happens in the next stage called diplotin stage, the fourth stage of prophase 1 of meiosis 1. The release of chiasma and the separation of homologous pair from crossing over will be complete by the diakinesis stage. So this process of separation and detachment happens in diplotin stage, and by the beginning of diakinesis stage, there will not be any crossing over or any chiasma. So in the diakinesis stage, what happens is the nuclear membrane will start to disintegrate. Okay, the nuclear envelope. The same with the prophase 1, what happened in the mitosis. Similarly, the nuclear membrane starts to disintegrate. By the end of diakinesis, the prophase 1 of meiosis 1 comes to an end. So what happened in prophase 1? Let's have a quick recap and compare it with what happened in the prophase of mitosis. You remember, in prophase of mitosis, the chromosomes, they just started to condense, and the microtubules started to develop, and the nuclear envelope started to disintegrate. But in the case of prophase 1 of meiosis 1, the genetic material DNA in the form of chromosomes, they crossed over. They went over a process of recombination. They just made a new variety of new possibility of chromosomes from the existing chromosomes genetic material. This crossing over enables variation. Remember, the mitosis, ultimate aim of mitosis is to create identical cells, but the ultimate aim of meiosis is to create variant cells, gametes that are different from each other, to produce variation in a population. So that is why meiosis once prophase 1 is having this crossing over process. This crossing over process ensures that variation happens in a population, or else all of us would be looking just like our parents. There won't be any difference. So this meiosis process makes the difference possible. Okay. So what happened in the metaphase of mitosis? In the metaphase of mitosis, all the chromosomes aligned in the metaphase plate to be pulled across the pole. Okay, to be pulled towards the pole, they arrived on the metaphase plate in the metaphase stage in highly condensed form. Similarly, the chromosomes in metaphase 1, they arrange in metaphase plate, but they don't arrange in line. Rather, they'll arrange themselves as pair. Okay, you remember, the homologous chromosomes, they will pair together and they will stand in a line as pairs in the metaphase plate. Okay, now the spindle fibers will attach to themselves and they'll start to pull. Instead of the sister chromatids, they'll try to pull the pair away. So now we are trying to pull the husband and wife, not the conjoined twins. Remember, in the metaphase of mitosis, the ultimate aim was to pull the sister chromatids apart from the centromere that is joining these two sister chromatids together. But In the case of meiosis, we want to the cells want to pull the chromosomes apart, the homologous chromosomes apart, 23 this side and 23 that side. We are not pulling the sister chromatids. The sister chromatids still stay together, but instead of that, the pairs are pulled apart to the other side of the pole. And this pulling process will intensify in the anaphase 1 of meiosis 1. In the anaphase 1 of meiosis 1, unlike the anaphase of mitosis, where the sister chromatids being pulled apart, but in the anaphase 1 of meiosis 1, the homologous chromosomes, they are separated. The husband and wife get separated instead of the conjoined twins. So this separation process goes on till the homologous pairs are separated, 23 this side and 23 that side. So after this, we'll have the telophase 1 of meiosis 1, where the chromosomes that went this side will decondense and the chromosomes that went that side will decondense, and we'll have the nuclear membrane will now start to appear, resulting in two nucleus inside the same cell, and the cytoplasm, it will have a depression, it will have a furrow, and this furrow, this cleavage will develop and it will result in cytokinesis, similar to the mitosis. But here we are having 23 chromosomes. Unlike the sister chromatids, these homologous chromosomes that went this side will only be carrying half of the DNA, not the entire DNA. The sister chromatids are still with the chromosome, but they are not pulled apart. Only the homologous pairs have went this side and that side. So which means we only have 23 centromeres. As I said, chromosomes are counted based on the numbers of the centromere, which means we only have half of the genetic material here, half of the genetic material here. It is safe for us to say that these two cells are now haploid. By the end of cytokinesis, after the telophase 1, we'll have two haploid cells, and that marks the end of meiosis 1. After meiosis 1, we'll have two haploid cells. These two haploid cells will undergo further cell division, which we call it as meiosis 2. Meiosis 2 is very similar to mitosis. The goal of meiosis 2 is to pull the sister chromatids that are present inside the haploid cell, which resulted from meiosis 1. Now these two haploid cells are going to divide. If we remember correctly, before going into the prophase, the DNA will replicate. We saw that in mitosis, so that when the chromosome emerges, it will have sister chromatids. Okay, this doubling of DNA is not done in the prophase 2. If we remember correctly, in the meiosis 1, the sister chromatids weren't pulled apart. Okay, they were already in the cloned stage, and there is no need for cloning any further. So that is why there is no doubling of DNA before the prophase 2 of meiosis 2. Okay, in this prophase, what happens is the chromosome starts to condense, and then the microtubule starts to develop out of the centrosomes, and then the nuclear envelopes start to disintegrate. This marks the end of prophase 2 of meiosis 2. After this, the cell will go through metaphase 2. In metaphase 2, what will happen is we have remember, we have 23 chromosomes, 23 centromeres with sister chromatids. These chromosomes will order and line up in the metaphase plate, similar to the mitosis metaphase. In this metaphase, the microtubules will try to connect to the kinetochore, and once they are connected, they'll start to pull. This marks the end of metaphase 2. After this, the cell will go through anaphase 2, which is again similar to the mitosis, where the pulling happens and the sister chromatids are pulled apart. Remember, in meiosis 2 anaphase 2, the sister chromatids are being pulled, whereas in the meiosis 1's anaphase 1, the homologous chromosomes are pulled. Okay, now once these sister chromatids are pulled apart, there will not be any duplicated version of chromosomes. There will be only chromatids going to the polar region. Okay, once they reach the polar region, that marks the end of anaphase 2. After that, telophase 2 happens, in which the nuclear envelope start to develop and the chromosome decondenses. By the end of telophase, we will have the furrow which will develop, and cytokinesis will happen, resulting in two new cells from that one cell of meiosis 1. So we remember we got two cells from meiosis 1. These two cells will divide and will give rise to four haploid cells. These four haploid cells are nothing but gametes. So now we got gametes. These gametes will go and fuse with the other gamete to produce a zygote. So this marks the end of meiosis 2, and by the end of meiosis 2, meiosis is over.